Thank you, everybody online and doing remotely, and thank you, everybody in person. Actually, more than I expected. So thanks for making the trip over. I'm Matt Scarborough. I'm an assistant professor in civil and environmental engineering. I've been at UVM about 3.5 years, and so I'm going to share a little bit of our work. So as was alluded to, I do a lot of work with biological processes to deal with waste and recover beneficial products from waste. And so I'm going to focus on just one of those ways. I'm going to focus on manure, which we've been thinking a lot about. And thinking about sort of this paradigm shift where we're thinking about manure as a waste, and taking that and actually treating manure as if it were a co-product that has some value that we can recover. And so that's sort of the journey I'm going to go on today. I do a lot of microbiology research, and so my talk today, we're going to start kind of big picture, dive a little bit into the weeds, hopefully not too much, and then we'll come back up and sort of end on a big picture. But if microbiology is your thing and you want to talk nitty gritty stuff at some point, I always welcome that. And so I'll be avoiding it for the most part today. So actually, let's go back. I don't want to do that. So I think you're in Vermont, you all know manure. You know what manure is. And sort of, I'm just curious, like, what you might think about. You don't have to tell me, and you don't have to close your eyes. But if you imagine manure, like, what do you think of? Smell. Pastures. Cows. Horses. Great. Thank you for playing. And so in Vermont, a lot of the manure that we generate on farms ultimately ends up in something that looks like this, a manure lagoon or a manure pit. And so this is actually a picture from one from New York, sorry, but it actually tells you what it is. So usually you'll see these facilities, and you know, we have a barn, and there's typically mechanical scrapers and things in the barn that are going to convey the manure in the bedding into a pit that looks something like this. It sits in this pit, and then what happens is manure eventually it gets spread on fields. And so they use this and they recover some of the nitrogen and phosphorus that's in here, and there's some agronomic value to this material and spreading it on fields. Of course, as you all know, if we put too many nutrients in one place, the Champlain Valley, for instance, we can end up with issues associated with eutrophication. And so one of the challenges of this sort of lagoon-based process, there's a couple issues with this you might imagine. And so they smell, that's true, but they also make greenhouse gases. And so in these processes, if it's converting into an anaerobic environment, we get methane production and release from these pits. And there's also in this product, this manure, if we're thinking about it, trying to think about it as a product, it's a fertilizer, but it has fixed nitrogen, phosphorus concentrations, not always necessarily at the right sort of ratios that we might need for certain areas if we're going to use it as a fertilizer. And so what I think about is what can we do with manure besides put it in a pit? And so anybody heard of ways that we also deal with manure? Anaerobic digestion, right? And so I'm going to spend some time introducing anaerobic digestion. And I want to show this facility. So this is what an anaerobic digester looks like. So you can kind of imagine it as you take this lagoon, you create a more sort of controlled environment. And now instead of producing greenhouse gases that are fugitive emissions from a manure pit, you're actually you want this thing to go anaerobic. You want to create an anaerobic environment where you're actually getting production of what we call biogas. And so biogas is a mixture of CO2 and methane, and it has value in that it can be combusted and used to generate electricity, or it can be cleaned up into renewable natural gas and be put into a renewable natural gas pipeline. And so this is the famous digester in Salisbury, Vermont, on Goodrich Farms. It opened up to a lot of fanfare. Dr. Garamello was down here at some point for a big ribbon cutting. And so this is the facility. I just want to be clear that there's between 14 and 15 on-farm anaerobic digesters in the state of Vermont. And they vary in a lot of sizes. They all have unique challenges and issues. but a lot of these were built between like the year 2000 or 2002 and like 2010. And so there are other facilities. I just want to show you this picture of this one. And so this is what an anaerobic digester looks like. And so these big tanks where you're getting biogas production. And the other nice thing about these facilities, you can recover biogas. And so I think of these facilities in sort of a multi-stage process, multi-phase process. And so at this particular facility, there's manure coming in that's going through a screw press, and the farm is actually recovering bedding on the front end, so they can reuse this bedding as material for their farm. And then that liquid stuff from the press is going into the anaerobic digester. This farm, as many farms do, they also do what we call co-digest. They get other organic rich substrates, in this case, a lot of food waste and food processing residuals, and then they also feed that into the anaerobic digester. This is a little different because they have this hydrolyzer tank, this separate biological process where they're pre-fermenting this food waste before they put it into the anaerobic digester. And so, again, anaerobic digester is producing the biogas. I just want to highlight some other neat things we can do. And so this gives us a nice way to start processing this liquid as well. And so this material that goes through the digester, we have the gas and we have the liquid residual. This liquid residual can be treated to actually separate out phosphorus-rich materials from nitrogen-rich materials. And when you do that, you're now separating your nutrients and giving farmers more flexibility in sort of how they would apply nitrogen and phosphorus. And so by decoupling this, you have more control over the ratios that you might be applying. And so my main focus is actually on the microbial communities that do this work. And so I study the microbial communities in these tanks and similar systems to try and convert organic waste into different products. Sorry, I got ahead of myself. Sorry, last thing. This particular facility, what they're doing with their biogas is they're actually putting it through a cleaning and scrubbing system to remove the CO2. They end up with an essentially renewable natural gas. It's like 97% methane. This goes into an existing pipeline. And actually, they had a contract with Middlebury College, and Middlebury College sort of pays for this specific gas. Again, this is one example. What this facility has in common with the other facilities is that they're all producing biogas and trying to make as much biogas as they can. This is different from every other digester in Vermont, and that every other digester in Vermont generates heat and electricity and doesn't actually produce renewable natural gas. This is a really attractive option if you can, but we're in Vermont and there's a lot of places where you're nowhere near a renewable natural gas pipeline or a natural gas pipeline. And so regardless though, two things that we're interested in doing, sort of our quest in our lab, make more methane quickly and reliable. And so it makes people nervous talking about producing methane, but it's methane that we want to capture as a renewable fuel. And so if farmers are going to be doing biogas production, we want to enable them to make as much as possible, to generate as much energy as possible. And we want it to be done reliably. And so these are living ecosystems that we have to manage and keep happy in order to keep biogas production going. And so we're trying to help them do that. And then the other thing we do is, what if we don't make methane? Is there anything else we can make that would be more beneficial than producing energy? And so that's a big question that we're exploring in my lab. And so our approaches, we study bioreactors of multiple sizes. So we have, in our lab, we have little tiny chemostat reactors that are 100 milliliters. We have some lab scale 2-liter reactors, all the way up till we go out and sample from full-scale digesters. And that's kind of our thing. We like to study these systems at different scales. We apply a lot of multi-omics tools to actually characterize the microbial communities that are in these systems, to understand the roles, the functions, the redundancy that is in these communities, and how we can sort of enrich for different activities based on sort of the genomic potential and what these organisms actually do. We try to model microbiome behavior. So we use multiple approaches to do that. I'm not going to talk about that today. And I also want to point out that we collaborate with farmers and other smart people. And so there's a lot of issues that go into this. And so I have a project right now that I'm not really going to talk about, but I have a project with Dr. Eric Roy, funded by the EPA, where we're literally going out and talking to farmers, and that are trying to run digesters, and it's been it's been an incredible experience. And so talking to farmers about their needs and working with them to sort of come up with some of these more advanced manure management practices. So I want to focus on this first sort of task that we're after. And so I want to give an example of some of our work. It's a metagenomic analysis, and I'll show you why we did that and sort of what we learned from it. But this is from a full-scale digester. It's separate from the digester I've introduced, but this work was done by Amy DeCola, who just graduated from the lab. And there was this full-scale digester starting up, and we kind of hooked up with the people that were starting it up. And from the day they started putting manure into this brand new digester, we went down every week and started collecting samples. So Amy went down and collected samples for an entire year from multiple tanks as they sort of came online. And we wanted to see how the microbial community sort of adapted, came together, established, and how resilient it was in this reactor. And the idea being, sometimes it can take people up to a year to really start up one of these systems. And the general convention is you just go slow, and that's fine, it's fine, but we're trying to put some sort of thought into this and actually look at the microbial communities and see, okay, what is the microbial community actually, the core community sort of there? And so that's what we did. So in addition to sort of studying the microbiome, we were also able to get all this operational data from the digesters. And so when you're running a digester, the pH is important. So we have, we'll just focus on this line. Operating these digesters at near neutral pH is essentially where you want to be. And so we monitored the pH over time. The organic loading rate is how much sort of food you're feeding to the digester at any given time. And so you'll see it essentially increases, which is what you want to do. They're sort of slowly feeding things, let them adapt, let things build up, and then hopefully you don't upset anything. And what happens if you upset it is you get accumulation of an intermediate volatile fatty acids. We'll talk a little bit about those, but these are key intermediate. So you start with your complex organics, they're going to end up as volatile fatty acids before they ultimately end up as methane. And so what you want to do is you want to control this so that you're not getting too high accumulation of these volatile fatty acids. And you can see that they were kind of getting there, and then the VFAs came down over time. And then we're also interested in the methane, percent methane content. So again, biogas is mostly methane and CO2. And I'll tell you, this is one of the highest methane contents I've sort of ever seen in a digester, which I thought was really interesting. And so generally we like to be at 50% methane or higher. They were as high as 65%, sort of where they stabilized. And so I have to introduce some of the microbiology. And so we're going to go through it a little quickly. But essentially what's happening in one of these tanks is you're getting these complex monomers are getting broken down into their constituent units, or sort of monomeric units. And then these units are either going to be fermented to a whole different slew of fermentation products, including the ones you always think of, like lactate and ethanol. And then these compounds all need to end up as either acetate or hydrogen gas and CO2, because the methanogens, these archaea now that are going to produce the methane, they only convert either acetate or CO2 and hydrogen into methane. And so at some point we need to get all of these organics funneled into one of these key intermediates. And so this, I just want to be honest, this is the textbook. Like if you read a textbook on anaerobic digestion, this is the figure you're going to see. So I'm going to show you some sequencing results now that we analyzed. And so we took our samples, our biomass samples, and we did a metagenomic analysis where we sequenced all the DNA in these samples. And then we were able to use that DNA, the sequences, to assess sort of who's there, and then also look at specific genes of interest. And so we could see who's there, who they're related to, and also what's their genomic potential. And so first I want to show you who is there. And so this is just the potential methanogens in the anaerobic digester. And so each of these different colored bars represents a different genus of organism, including some unclassified ones. And so you can see a couple key points here. And so one, they added the inoculum. So they trucked in a whole bunch of sludge from a separate digester to try and kickstart this thing. They added that, and it took a couple of weeks, which makes sense because these are run at like a retention time of about 28 days. And so it takes about it can take up to a month to get like complete turnover sort of what's going on in the tank. And so we have this, they add the inoculum, and we see the abundance of these methanogenic archaea that we want actually plummet. And so they run it over time. And the other thing you'll see, this big red bar goes away. This big red bar is methanocorpsiculum. And if you ever want to know anything about methanocorpsiculum, it's a methanogen that we know is in cow manure. And so if we go sequence cow manure, we almost always see it. And we almost never see it in digesters. And so we're not really sure why, but that's certainly the case for what happened here. What we do end up with is methanogens that belong to essentially this family of methanomicrobiales. Even these other genuses fall within this family. And all of those organisms make methane in one specific way. They make it from CO2 and hydrogen gas. And so I showed you two routes before. The ones that are using the acetate, they're just barely even there. They're barely even there. And so we could dig in a little further. Oh, sorry. We could dig in a little further and actually look for specific genes. And so this is just one example of sort of what this data looks like. And looking at this, there's specific genes of interest, and we look at the abundance, and now we're looking at the two different tanks. So we're looking at the anaerobic digester separate from this upstream hydrolysis tank. And without getting into the function and the pathways and all that, you can see that there's differences. And based on these differences, especially this gene here, this XFP, which is it's a key component of this specialized sort of glucose degradation and xylose degradation pathway, the bifid shun, if you're curious. And then these abundance of all these genes associated with producing lactate, acetate, and ethanol are much higher abundance. The other thing we notice is propionate production genes are essentially absent from either of these, which propionate is this 3-carbon carboxylic acid that accumulates in a lot of reactors that are failing. And so we just don't have organisms really that have the genomic potential to produce this stuff to begin with. And so one of the take homes here is this hydrolysis tank, remember, that's feeding right into our digester. These fermenting microbial communities are very, very different, very different. So what does it all mean? So this is sort of the conventional diagram, and this is what we're seeing. And so we have two separate tanks. And so in this full-scale hydrolyzer, the whole point, it's called a hydrolyzer. It's intended to kickstart this hydrolysis process. And it's doing that, but it's also fermenting, which we'd expect. And so it's doing hydrolysis, but what we found is essentially all the fermentation pathways are associated with this acetogenesis route. And so there's acetate being produced, lactate and ethanol are the 3 primary products being produced. And so butyrate, propionate, and some of these other sort of more complex carboxylic acids, we didn't measure them, and the genes aren't there to produce them. And so if we look then into the full-scale anaerobic digester, it's also sort of different than the sort of textbook that we would expect, in that we're getting production of these intermediates. This acetate, lactate, and ethanol from the hydrolysis tank is ultimately being converted into butyrate and caproate, very likely. And then those acids are getting oxidized to produce CO2 and H2. And the main route of methanogenesis in this reactor is believed to be through converting CO2 and H2 into methane. So if you have any detailed questions about that, we can talk about it. But I want to also hit on our other sort of major goal as a lab, and that's to make products that aren't methane. And so methane, it rightfully has a bad rap because a lot of it we get from fossil fuels, and it's kind of as a product. When you start talking about refining, it's a really low value product. If you're just going to put it in a combustion engine, like you're not going to get that much money for it. That's sort of how this works. And so if you put your chemical engineering hat on, you start to think about what else can be produced. And so what my lab spends a lot of time trying to produce in our lab now is medium chain fatty acids. And so we're interested in producing primarily hexanoic, which is a 6-carbon compound, and octanoic acid. And why? Because they're valuable, because they're about 1,000 times more valuable on an electron basis compared to methane. And so there's a huge demand for these. I won't get into all of them, but they're currently derived from palm refining. And so there's a way to do sustainable palm refining, we think. But by and large, I think some of you are cringing. And so, okay. And so they're relatively easy to recover and purify. And so compared to some other compounds, they're really insoluble. So it's kind of nice because if you produce them, you think about producing biogas, that's great because it just sort of comes out of the tank as a gas and you can capture it. These compounds, they're still liquids, but you get enough of them, they tend to float to the top. So you can sort of just skim them off, hopefully. I wish it was that easy. And then they're also platform chemicals. And so they're used to produce a lot of things, including liquid transportation fuels, like conventional gasolines. So if you ever get the chance, go to a drugstore and you can look around. So my favorite use that I found so far of an MCFA, this is C8, right? Beard butter with protein. So if you ever get the chance, you find yourself at CVS, you can find these compounds in a lot of these sort of pharmaceutical compounds. All right, so right now what we're working on, we have lab scale reactors set up where we're looking at essentially using two different substrates to make MCFAs, medium chain fatty acids, where we have a student, Yota, focused on carbohydrates, and then we have two students, Leandro and Lucy, focused on protein degradation. And we're focusing on these separately, sort of understand separately how these things are broken down, if we can produce these compounds that we're interested in. And we're also testing a little bit how we can make synthetic mixtures of these. The idea being we could go out and find organic substrates that would give us some flexibility in the protein versus carbohydrate composition. And is there a way that we can use that composition to sort of drive production of some target? The other thing we're trying to make, we have a project funded by the USDA looking at propane. And so how do you convert manure to propane? And so I'm working with Dr. Bottaretti and Dr. Andrea Hicks, actually at the University of Wisconsin, to do life cycle assessment associated with this work. But we're setting up reactors, by we I mean Mona, to actually take manure, ferment it into butyric acid, and then use that butyrate and use a photoreforming reactor that relies on a catalyst and light to actually turn this carboxylic acid into what you end up with as byproduct is hydrogen and propane. And so we're studying the microbial communities, and then Dr. Bottaretti, he's a very talented physical chemical processes person, is working on that step of the process. And so that's all I want to say about propane production. And I want to end though going back to a really big picture. And so we talked about manure. And so I, as a lab and sort of as a researcher, what I'm really interested in is understanding how we take things that we generally refer to as waste, organic waste, even gaseous waste. Even on a mass basis, we generate a lot more gaseous waste than we do solid waste. And so my main interest is understanding the microbial communities and sort of how we can sort of engineer and influence these communities, sort of derived from nature, but sort of just tweak them a little bit and push them to make certain products of interest. And so everything I've talked about today is, and if we think about what we need as a society, there's classifications of different chemical compounds we're just going to need if we're going to exist. And my hope is that if we get enabling biotechnologies, all we need are some organic waste, maybe some trees and some grass and some gaseous waste, and we can start to expand production of all these different chemicals that a society might need. And the driver here is, again, to repurpose waste, one, and two, by doing this, you just provide more equitable access to all these things that a community needs. You don't need to be sitting by a palm refinery, and you don't need to be sitting on a petroleum reserve in order to sort of kickstart all this manufacturing. And so that's my motivation. And so I just want to thank the funding agencies. And so NSF has funded a couple of projects. The US EPA is funding me and Dr. Eric Roy on a project where we're getting to go out and talk to farmers. The USDA is funding a project looking at propane production. Cabot, we're helping Cabot actually figure out what to do with some of their wastewater and see what we can recover from it. And then Vermont NASA EPSCoR is also supporting some of this work. And so the people that actually do the work, and so this is the latest iteration of my lab. And so thank you, Kennedy, you're here. Leah, you're here, thanks. And so you saw some of these folks earlier. I just want to give another shout out to Amy. She just graduated. She's a wastewater process engineer now. And she did a lot of the work at the full scale digester, and I presented her data today. So with that, it's 12:30.
You crushed it.
And I'll take any questions you might have.
One of the waste streams that like I